Jai Gurudev children. Today we will discuss about the matter phase 1. So already we have studied about the prophase 1, where we have studied about the 5 stages. Those were the leptotin, zygotin, pectitin, diplotin and dikinesis. Now after that the next phase is the matter phase 1. So in matter phase what happened? That now the chromosome are scattered in the cytoplasm soon after completing the prophase 1. The nuclear membrane also disappear, nucleolus disappear, and now the chromosome remains scattered in the cytoplasm. Now although in the prophase 1 we have studied that the homologous pair, they try to separate, but although the separation takes place, they are not separated from one another. They remain nearby. And those chromosomes, that means that you can say the tetrad, now again they occupy the equatorial region of the cell. Just like in the mitosis we have studied that in the matter phase the chromosomes, they are arranged on the equatorial plate. So similarly in the meiosis also the chromosomes are arranged in the equatorial plate of the cell, and each of the chromosome, they are connected with the help of spindle fibers. Now you just see, just to explain that, here already the diagrams are given. You just see here. So this one, the matter phase 1, here you just see the chromosomes which were scattered in the prophase. Now they are arranged in the equator region of the cell, or you can say in the center of the cell. Now here we were discussing while discussing we have used the 4 chromosome. So here also they are having the 4 chromosome, but you just see they are in group. They are in group which is known as the tetrad, which already we have studied about the tetrad. So in this form the chromosomes are arranged in the equatorial region, and each of them, they are connected by the spindle fiber, and the spindle fibers are connected, means they join one particular chromosome in the kinetochore. So which already we have studied, kinetochore means the plate which is present just near the centromere. So like that what happened here, 2 chromosomes are there, each of them, they are connected by the spindle fiber. So this change, it occurs in the matter phase. Now if you see, you can see the difference between the mitosis. In mitosis what happened, that if they are having 4 chromosomes, they are arranged in a single line. But here, although they are having 4, but in the form of tetrad, they are present. So in this way it differs with that of the mitosis. Now soon after that, the next stage is the anaphase 1. Now in anaphase 1 what happened? Now here they were in the form of in group, in pair, or in the form of tetrad. Now this tetrad, it separates, and one chromosome, they move to their respective pole. So you just see, if you compare this diagram and this, the distance between the 2 chromosomes had increased. Here it was much less, but here it increased. That means both of them have repelled, and after repelling they are moving to their respective pole. So here also one difference is there with that of mitosis. In mitosis what happened? Each of the chromosome, the centromere was splitted, but here the splitting doesn't take place since they are in the form of pair. So one whole chromosome moves to one pole, whereas the other whole chromosome, they moves to the other pole. So just the separation of that homologous pair takes place in the anaphase 1, and in that way it differs with that of anaphase in mitosis. Now after anaphase, the next phase is the telophase. So in telophase, what happened? The two chromosome, means here's two chromosome I am talking because I have used two chromosome. Initially I have taken 4, now they are having 2. So so that's why I am mentioning as 2, but it may differ in different organism. So now the two chromosome have reached their respective pole. After that, the formations or the appearance of the nuclear membrane, nucleolus, had taken place. So when they have reached their respective pole and the formations of the nuclear membrane and the nucleolus takes place, it indicates that they have completed the last step of the karyokinesis, that is the telophase 1. So this thing, it happens in the karyokinesis of meiosis 1. Now after that, soon after karyokinesis, the process is not completed. After that, some changes occur in the cytokinesis. So cytokinesis, already you all know, that is the divisions of the cytoplasm. And what happened? That single cell which we were talking about, this single cell, now after that the division of the cytoplasm takes place. And the division, it differs both in case of plants and animals. In plants, it starts from, in animal cell, it starts from the external part to the internal. And if it is plant cell, inside one wall-like structure develop which extend in both the direction. So that same difference occurs even in here, but in the cytokinesis, the division which it takes place is the means the divisions of the cytoplasm. So soon after cytokinesis, what happened? That single cell, it is divided into 2 cells, each of them having their own nuclei. So once when the cytokinesis is completed, that means they have completed the first meiosis division. But here you just see, at the end of cytokinesis, no doubt 2 cells are formed, just like the mitosis. But here if you see, each cell, they are having 2 number of chromosome. But in the parent cell, they were having 4 chromosome. So that 4 has been reduced to 2. That means the reduction division had taken place. So now I think it's clear how the reduction division takes place. And one more thing, children, you all should know that this reduction division, it takes place in the meiosis 1 actually. In meiosis 2, the reduction division doesn't take place, because once when they have entered in the meiosis 2, whatever the number of chromosomes they are going to have, even after forming the 4 cell also, they are going to have the same number. So the second meiosis division, it is similar to the mitosis cell division. So that much you all have to remember. But before that, one thing, now once when they have completed the cytokinesis, now these 2 cells, they haven't completed the meiosis division. They have to undergo the second division, that is the meiosis 2. But before going to the meiosis 2, these 2 cells have to prepare themselves to undergo the meiosis division. So that means what? These 2 cells also will enter a non-dividing phase. And we have studied, when we talk about non-dividing phase, we now know it is known as interphase. But children, in case of meiosis, after completing, in case of meiosis, that means after completing the meiosis 1, the cell, when they enter into the non-dividing phase, it is known as interkinesis. Now here some difference are there with that of the interphase. So there what happened? This, in the interkinesis phase, the 2 cell, means they will enter into the interkinesis phase, where they will undergo G1 and G2 phase. That means G1 plus G2 gives is the result, or is also known as the interkinesis phase. So in both the cell, the interkinesis process will take place where the S phase is absent. Now the question may arise, why this S phase is absent? Now already now you all have the idea. S phase means what? There the replication of DNA takes place. That means the DNA materials are double. But here you just see now already, and DNA material double means what? I have said that initially a particular cell inside the nucleus, if they have one chromatid, you just see if they have one chromatid or chromatin, they gets double, and this doubling of this one, it takes place in the S phase. But here if you see, here already they are having 2. So, and we know that chromosome means they have 2 chromatids. Now in this 2 cell, they are having the 2 chromatin. Here again they need not have to add more chromatin. So already they are having, so if they have to add, then if it has to be added here, that means the chromosome structure will be little bit different. So as they are having this 2 chromatin, that's why in them this S phase doesn't take place. And since the S phase is absent here, that's why here that's non-dividing phase, it is known as interkinesis. So now I think it's clear why this particular phase doesn't means this S phase doesn't takes place, or what is the difference between interkinesis and the interphase. So soon after completing the interkinesis, that means after completing the G2 phase, the cell, this 2 cell will enter into the meiosis 2 cell division. So I think, so you just see here the rough one I have shown. Now if you see here in the board, if you see the second meiosis division, now here also just like in meiosis 1, first the karyokinesis will take place, after that the cytokinesis. So now here I have taken, now here what happened, first stage, that is the prophase 2. Now in the prophase 2 what happened, now one more thing, this phase is similar to mitosis. So here the means the whatever the changes takes place, it's almost similar to that of mitosis. So in the prophase 1 what happened, the chromatin materials again is converted to chromosome. So and the nuclear membrane which were present here, now this nuclear membrane is disappear, and now the chromosome remains scattered within the cytoplasm. So this thing, it happened in the prophase 1. Now soon after prophase, not prophase 1, in the prophase 2. Now soon after prophase 2, they enter the metaphase 2. So in metaphase 2 what happened, now these chromosomes, now here one thing children, here I haven't drawn both the cell, only one cell I have used. So same thing will happen in both the cells, simultaneously it will take place. So in the metaphase stage, what happened? The chromosomes now they are arranged in the equatorial region, or in this metaphase plate-like structures. And each of them now they are connected by the spindle fiber. So this thing, it happened in the metaphase 2. Now after metaphase 2, the next phase is the anaphase. So here what happened? The splitting, you all know already, the chromosome means what? They are having like this the chromosome structures with this centromere, and this centromere it splits. So after splitting, now they are having 2 daughter chromosome, each of them they are having one chromatid. So that means what? Here the splitting had taken place. After splitting, now this daughter chromosome, they try to move in their respective pole. Now here the diagram properly I haven't drawn. So that means here you just see now one chromatids they are having, and they are moving to their respective pole. So this thing, it happened in anaphase 2. Then it enters into telophase 2. So in telophase 2, the daughter chromosome reach their respective pole, and now the appearance of the nuclear membrane takes place. So the nuclear membrane appear, even the nucleolus to appear here. After that, the division of the cytoplasm takes place, which is known as cytokinesis, and ultimately this one cell, it is divided into two cells, and each of them, they are having the nucleus with the chromosome. Chromosome means what? Here one difference, each of them, no doubt they are having two, but they are having only one thread like structure. So in this way, ultimately from one cell it has given rise to two cells. But since in the meiosis two cells are there, so at the end four cells are formed, and each of the four cells, they are going to have two daughter chromosomes. So these are the four cells or the four gametes which are formed after completing the whole meiosis division. Now if you see, already I have said, like if you see in the meiosis 2, if we take meiosis 2, these are the parent cell. So here they are having only two chromosomes, and at the end also they are having two chromosomes. So that means what? Here when you compare this and these cells, they are having the same numbers of chromosomes. But if we think about the original one, original means the parent cell which had started the meiosis division, that means which had started the meiosis 1, at that time they were having 4 chromosomes. So at that time they were having 4 chromosomes. So this 4 chromosome only, ultimately it has been reduced to 2. And this reduction division had taken place in the meiosis 2. In the meiosis 1. So the reduction division, actually it takes place in the meiosis 1. But although in the meiosis 1 takes place, that same thing, that same numbers of that reduction which had taken place, that means the 4 new chromosome had been reduced to 2, and at the end of the meiosis 2 also, each of the cell, that means the 4 cell which form, there also they are having 2 chromosomes. So that's why the whole process, it is said to be meiosis, it is said to be the reduction division. But if we compare only meiosis 2, it is similar to that of the mitosis division. So this much you have to remember. Now mainly from this part or from this topic, certain thing it should be clear. One is the crossing over, when the crossing over had taken place, or in which stage the crossing over starts and in which stage it is completed, and what happens in the crossing over. Beside this, what is synapses, you have to know. What are homologous chromosome? Then why the meiosis is known as reduction division. So if these things are clear, then easily you can attempt any question, whatever it is asked from this chapter. So if those concepts are clear, easily you can attempt. Even the diagram also you have to practice from this part. So mainly the diagram means what? How the crossing over takes place. So that diagram is very important. Beside this, you have come across the term synaptonymal complex. So what is synaptonymal complex? What is tetrad? So those things, it has to be clear. Then only you can attempt any question you are asked. So this much for today. Thank you. Jai Gurudev.